Hi, everyone. We'll just give folks a moment or two to join, and then we'll get things started. Okay, welcome everybody. I'm Jeff Kahn. I'm the director of the Berman Institute of Bioethics. For those who don't know me, it's my pleasure to welcome everyone to another of our bi-weekly Berman Institute seminar presentations. I should say before we get started and I hand things over to my colleague Deborah Matthews to make the introduction, that today is the final seminar in the academic year 2021. And so I need to say a few thank yous. One, thank you all for joining during the remote period of the pandemic. It's been really great to have such good attendance and great discussions. But it's really, I think, important that we point out that these seminars have happened because of the people who have organized them and done the work behind the scenes. So first, let me say big thanks to the BI Seminar Planning Committee, chaired by Joe Ali and joined by our colleagues Alan Regenberg and Mary Catherine Beach. So applause to the group for all the work they have done in getting such a great lineup of speakers. And maybe even more important, a huge thank you to Suzanne Kneisner, who's done all of the work behind the scenes to get our speakers set up and everything to have gone flawlessly, and the seminars to work as they do via the webinar function and all the things that look easy but are really not. So thank you, Suzanne. Tons of work behind all this, we know, and we appreciate all you've done to make that possible. So we're going to take a summer hiatus and we'll be back in the fall, of course, and all of our lists will receive information about the calendar when that's available. So stay tuned for that. Okay, without further ado, let me say welcome to Malia, who you'll hear about just in a moment. And we're really thrilled to have her today joining us for seminar. I'm going to make the introduction of both Malia and the topic. I'm going to turn things over to my colleague, Deborah Matthews. So Deborah, over to you.
Thank you very much. I am delighted to have the opportunity to introduce Malia Fullerton, who I have now known for over 20 years. Malia and I share that we are trained geneticists and then transferred over to the bioethics world. She, I guess it was in '98, joined the department at Penn State, where I had done research as an undergrad with Ken Weiss, who subsequently introduced us because of our sort of similar interests. And I've known Malia and respected her work ever since. So Stephanie Malia Fullerton is a professor of bioethics and humanities at the University of Washington School of Medicine. She's also an adjunct professor in the departments of epidemiology, genome sciences, and medicine, and an affiliate investigator with the Public Health Sciences Division of the Fred Hutchinson Cancer Research Center. As I mentioned, she got her doctorate in human population genetics from the University of Oxford, and then retrained with a grant from the NIH in the ethical, legal, and social implications of genetics and genomics. Dr. Fullerton leads the ethics teams of multiple large genetics projects and consortia, and we are delighted to have her join us today for the Berman Institute seminar series, on which she'll talk about diversifying precision medicine, reflections from the trenches of clinical translational genomics. Welcome, Malia, and thank you so much for being with us.
Thank you so much, Deborah. And my gosh, yes, it has been over 20 years now, which is just incredible. But anyway, thank you so much for that very kind introduction. I am delighted to be here. I'm going to tee up my slides. Try to get going here. Okay.
Looks great.
Great. Wonderful. Thank you, Suzanne. And thank you again, Suzanne, for your help with this presentation. Yeah, as Deborah said, I'm going to be talking on this topic, diversifying precision medicine. And I am going to be talking from my vantage point as someone who's been directly involved in a number of the emerging clinical translational genomics studies, particularly in my capacity as an embedded ethicist. And but I will try to kind of pull out, for those of you who may not be coming at research ethics and research ethics concerns from a primarily genetic lens, although that is how I spend my days. Okay, I got to make sure I can move forward here. Okay, before we get started, I will just begin, as I always begin my presentations, by acknowledging that where I sit here in my home, and then more broadly, the University of Washington campus sits on lands that have been occupied, that were the traditional home of a number of tribal communities, including the Tulalip, Muckleshoot, Duwamish, and Suquamish tribal nations. Without those folks and that land, we would not have access to working, teaching, and learning environment. And I just want to humbly and gratefully acknowledge that and thank the original caretakers of this land who are still here. Land acknowledgement is essential. And of course, it is not enough. And there are ways in which to pay back for some of past injustices, and we try to do so here at the University of Washington. So now that I've gotten that out of the way, I'd like to begin by telling you how I would like to leave you at the end of my presentation today. And actually, I'm going to start my timer because it's the first time I'm going through some of this material. I want to make sure I don't lose track of time. I'll begin by discussing what I've described, and I think many people recognize, as the current diversity problem in much precision medicine, and indeed a lot of biomedical research. I will then step through some case studies of recent precision medicine initiatives, which are illustrating some of the challenges entailed by attempting to do our work more inclusively. And these challenges are going to fall in three main areas in my discussion: challenges involved in managing diverse data, challenges involved in engaging with diverse stakeholders, and challenges involved with intervening on diverse risk factors. And I'll kind of step you through those and some of my reflections on those from my in the trenches perspective, and then spend a little bit of time, if I don't lose track of time, at the end of my presentation, addressing the ways in which we might move forward in the face of these challenges in order to enable precision medicine and clinical translational genomic approaches which will benefit all people. So let's start with the problem. The problem really can, of course, be traced in its genesis to all of the current precision medicine programs can be traced, of course, to the successful completion of the Human Genome Project, that 10-year and $3 billion international effort to decode the nearly complete sequence of a single human genome. That scientific success provided the basis of a purported revolution in medicine, one in which individual level genetic and environmental risk factors could, should, and would be employed in the promotion of public health in a manner that would be preferable, as Francis Collins noted here in one commentary that he wrote after the completion of the Human Genome Project, preferable to the traditional one-size-fits-all approach of identifying disease and intervening on disease risk. To achieve that objective of precision medicine, the National Institutes of Health and other biomedical funders soon began underwriting a class of genetic studies known as Genomewide association studies, in which first thousands and then subsequently now millions of gene variants in the DNA of patients and healthy controls were compared in an effort to pinpoint genetic risk factors underlying the susceptibility to various common complex traits. While this approach has gradually given way to more detailed genetic interrogations, including comparisons of exomes and now whole genomes, the GWAS investigation approach has proven an important workhorse of precision medicine research. Indeed, tens of millions of genomes have been interrogated in this fashion, leading to the identification of thousands of disease-linked risk variants. Unfortunately, the earliest GWAS studies were conducted almost exclusively in individuals of Western European ancestry. This was first brought to light by a study by Need and Goldstein in 2009. And a subsequent analysis led by a former student of mine, Alice Popejoy, and myself, suggested that the Eurocentric bias in this form of investigation persisted, improved somewhat, but persisted despite a nearly tenfold increase in the number of samples that had been studied between 2009 and 2016. So an increase from 1.7 million to 35 million samples, and still an overwhelmingly predominant bias towards the investigation of individuals of European genetic ancestry. These persistent biases in genomic investigation are concerning because, and this is where Deborah and my background in genetics, and in particular population genetics, comes in. They're concerning because although a great deal of genetic variation is shared within and among populations, genes do vary as a function of human demographic history. So an overfocus on individuals of primarily one population genetic ancestral background can limit the ability to generalize and apply risk information to patients from other parts of the world and other population genetic backgrounds. And this tendency towards affecting how the information can benefit individuals is especially problematic when we turn to a consideration of rarer variation, which tends to be more geographically restricted and hence population specific. Even before the biases in GWAS were noted, then director of the National Human Genome Research Institute, Francis Collins, published a prominent commentary in the pages of Nature calling for a nationwide longitudinal prospective cohort investigation designed to draw rigorous and unbiased conclusions, unbiased because the biases were already at that point recognized in the cohorts that were then available, about the roles of genes and environmental factors in health and disease. This call was actively discussed and debated, but ultimately the project idea languished in the face of budgetary constraints. And but as I think maybe most of us are aware, was finally launched by the Obama administration in 2015, and is now, of course, become known as the All of Us research program, and is actively underway and has already recruited many hundreds of thousands of individuals. There's the picture of the All of Us program. But before the All of Us program was launched, and in the interim, and with increasing urgency, of course, as the overwhelming lack of diversity in genomic discovery studies became obvious, a large number of precision medicine research initiatives designed to promote genetic research and its clinical translation began to be funded by the NIH. And these are just a few of them, and the ones that I'm going to focus on in my presentation today. These initiatives sometimes extended genetic investigation to previously recruited diverse cohorts. And I meant to say earlier that although diversity can be understood and should be, I think, appropriately understood and construed in many different ways, typically in the context of most of these genetic investigation, diversity tends to primarily be understood with regard to population genetic diversity, which then becomes in shorthand thought about in a recruitment context, in the context of racial and ethnic affiliations. And we can have a longer conversation about that, but I just wanted to make clear that that's how diversity is being construed in the context of these studies. These initiatives sometimes extended genetic investigation to previously recruited diverse cohorts. In other cases, they initiated new participant recruitment and even implementation of emerging tests and techniques. Many included some component of ethical, legal, and social implications, or ELSI engagement. Accordingly, I became involved in a number of these initiatives as a so-called embedded ethicist, and in that capacity have had the opportunity to observe firsthand some of the challenges entailed by recent attempts to incorporate more diverse data collections, patients and community members into what was once a kind of sleepy backwater, rarefied corner of academic medicine. So that is largely what I want to focus on today. I want to introduce you to a few short case studies that illustrate what I have come to see as a set of fairly important growing pains that are accompanying the current push to diversify precision medicine research and possibly all forms of biomedical research. And I will kind of reflect on whether and how these challenges will be transcended could have an important impact on how precision medicine is delivered and whether everyone who could potentially benefit from these approaches actually will. So I'm going to turn to my case examples here, and I'm going to go through three in turn, illustrating each of those sets of challenges that I briefly alluded to in my very first slide. So the first challenge I would like to address is the challenge of managing diverse data, or perhaps more appropriately, data from diverse populations. And I will illustrate this from my experience as a co-investigator with a research consortium known as Topmed, or Transomics for Precision Medicine. This is a research consortium funded by the National Heart, Lung, and Blood Institute. You can find more information about it at that website on the left, and a marker paper describing the work of the consortium was published earlier this year in Nature. And I'm a co-author on that paper along with about 400 others. So just very, very briefly, because it's a large and complicated project, just to kind of introduce you to Topmed as a research initiative. It began in 2014, and its initial focus was basically as a resource development project to add whole genome sequence data to studies that had previously been conducted where there were already in hand in the freezer samples and linked rich phenotypic information and other kinds of risk information, non-genetic risk information. It is currently in its 7th formal phase. Over 150,000 research participants from those previously collected studies have been involved now in the consortium, representing participants from over 90 research studies from both in the United States as well as collected internationally in various contexts. And it also involves the work of over 30 active working groups. So it's a very, very large group. The Data Coordinating Center for Topmed is housed at the University of Washington, which is how I got to be involved in this project. And there is a linked IRC, or Informatics Research Center, at the University of Michigan. Over the course of the last 7 years, there have been 7 different sequencing centers which have contributed whole genome sequence and increasingly other kinds of omics data to the project. So as of January of this year, whole genome sequence data have been generated for over 150,000 research participants of diverse genetic ancestries, and have been shared with the broader research community via the National Data Repository, dbGaP, the database of genotypes and phenotypes. And many of these incredibly rich and detailed and very kind of taking up a lot of space data are increasingly going to be moved to cloud-based analysis platforms. And for Topmed, that is going to be in the context of the NHLBI Biodata Catalyst cloud platform. Topmed is widely regarded as one of the most diverse collections of whole genome sequence participant data accessible and available, certainly in the United States context and possibly internationally. Just a very brief note, because I'm going to show you a number of these pie charts over the course of the presentation. This is an image that's from standard Topmed literature and how they introduce many people to the study. Notice for the people who are interested in how we talk about race, ancestry, and ethnicity, the very interesting way in which Hispanic, Latino, and ethnic category is called out alongside genetic ancestry groups. And there's complicated reasons for that I won't get into, but I think it's an interesting kind of interesting way in which Topmed chooses to describe and manage their data. There are many different ways in which I could describe and tell you about managing data in the context of Topmed. And I'm just going to focus on one recent example and some LC committee conversation about this example. and it involves a particular tool associated with the data collection called a Bravo server. And I'm sure Bravo is an acronym for something, but I don't remember what it is off the top of my head. But the Bravo server is housed by the folks, the IRC folks at the University of Michigan. And basically it is a place where investigators who want to have access to information about the genetic variation that has been identified in Topmed can go to this browser. The link is here. They can type in a gene name or a particular location in the genome, and information about that gene or SNP or gene location in the genome will come up if that region or that variable site is contained within the Topmed data set. And so the Bravo server provides information about chromosomal location, the different alleles that vary at a particular variable site, whether variations have any kind of functional significance, and importantly, what the frequency of these gene changes are, how common or rare they are in populations. And this is available for over 700 million variants that have been identified in over 130,000 deeply sequenced genomes. So it's a very, very important resource and rivals sort of other kind of more federally controlled resources, such as the DB-SNP database. Last summer, or maybe it was last fall, the Topmed LC committee, which I co-chaired, was approached by the Bravo server developers to ask if it would be appropriate for the server to report variant allele frequencies stratified on a population-specific basis in a manner that is common in other aggregate gene variant data lookup services. The pros of doing this is it would enhance, they argued, the research utility of this resource, allowing investigators who have identified gene variants in the context of other studies and particular population backgrounds to validate that the variants that they've identified have been observed in similar populations in other cases. It would also, they argued, facilitate assessment of the clinical meaning of gene variants. And this is becoming increasingly important as we are beginning to accrue more and more data in a clinical context, and we need to have reference lookups in order to interpret and understand the meaning of gene variants with regard to disease risk in patients. The cons that had been identified in a previous round of LC committee discussion, actually prior to my direct involvement in the group, had included potential for, if things were reported too specifically, the possibility that individual research participants could be re-identified, the possibility that people could mistakenly reify or understand population genetic variation as somehow undergirding or supporting racial biological distinction, and also the possibility that particular variants associated with particular stigmatizing conditions could be associated with particular specific populations. And so initially the plan with Bravo had been not to add these population identifiers or not to basically stratify the allele frequency reporting in this data set according to population background to avoid those cons or those difficulties. We began a conversation near the end of the year in the face of much other busyness. I stepped away from chairing the committee before the deliberations were completed, but my understanding is that the group has landed on a novel approach that would involve reporting allele frequencies stratified by genetic ancestry, and specifically stratified by a particular form of genetic ancestry, which is not based on the ancestry of an individual patient, but is instead based on the ancestry of a chunk of chromosomes surrounding the gene variant that we might want to report. It's a little bit too kind of in the weeds for me to explain why they landed on this approach, but the group specifically recommended this stratification with respect to ancestry as a way to get around some of the potential ethical concerns associated with ascribing gene frequency and other kinds of genetic information to particular social categories. And so it was adopted with a specifically anti-racist frame in mind. And it may very well be that that is the only thing that Topmed can do. However, the problem is that genetic ancestry is rarely, if ever, collected in the course of ordering a clinical genetic test. Instead, as we showed in a paper that was published in Human Mutation in 2018, test requisitions for clinical genetic tests collect a wide array, typically of social categorizations of genetic background, racial or ethnic identity, or some amalgam of the two. They're often contradictory, and they really do not have any clear relationship to a genetic ancestry estimate. So providing the Bravo server data stratified in this way might be the most straightforward way to present the data in the context of this large and complex precision medicine study. But is not probably going to help, in particular, the clinical genetic labs that really wanted to have this information to help them understand better the clinical meaning of particular variants they might be encountering in the context of clinical genetic tests. You know, and certainly, of course, we rarely have patients coming into a clinical setting with their ancestry so clearly written on their body, as shown in this kind of rather jokey cartoon here. Which is just to say that this was an anti-racist solution grounded in proper kind of genetic epidemiological practice, which may solve some problems associated with stratifying data in the context of Topmed, but will create others. What is the main take home? I think the main take home for me is that in the context of these large precision medicine initiatives, it's really hard. It's really, really hard when we have data coming in from many different places, many different populations, many different individuals of diverse population, genetic and geographic background recruited over many decades to organize, analyze, and report genomic data in ways that will allow us to do our work, bring data together, and create information that will ultimately be able to be applied in a precision medicine or clinical context. The second challenge I would like to discuss is a challenge of engaging with, is not a challenge of engaging with diverse data, but is rather challenges of engaging with diverse stakeholders. And I would like to illustrate this challenge that I think is coming up increasingly in a number of these precision medicine studies with regard to my participation in a different precision medicine research initiative, the CSER Precision Medicine Research Initiative, CSER being an acronym standing for Clinical Sequencing Evidence Generating Research. Like the Topmed Consortium, you can go to a website and learn more about CSER there, and a marker paper describing the work of the second phase of CSER, which is the evidence generating phase, was published near the start of the latest phase of the project in 2018 in the American Journal of Human Genetics. CSER is, in fact, a very, very different kind of a collaborative precision medicine research project than Topmed. Unlike Topmed, which was basically trying to create new genomic information for standing extant, in some cases been present for a long time, decades-long epidemiological cohorts, CSER began as a new initiative to collect and recruit in new people, and specifically began in 2010 with an initial focus to develop and disseminate best practices for integrating DNA sequencing into clinical care. That work continued for 5 years in the first instance, and then was expanded in a new round of funding explicitly to encompass and include more diverse, again, diversity primarily understood in terms of racial and ethnic diversity, and medically underserved patients. And that second phase began in 2017. Rather than there being hundreds of thousands of people sequenced, in this case, because we were recruiting new participants, patients who had a need for clinical genetic sequencing in various ways, the goal of recruitment has been to bring in approximately 6,000 participants. This is across six clinical sites across the United States, including one NIH intramural program, and includes 8 work groups. So it's a much more modest research program reflecting the need to kind of bring new people into research and to do so in an explicitly translational context, as opposed to Topmed, which was more at the basic science end of the spectrum. The coordinating center for CSER is at the University of Washington. The Data Coordinating Center is held there as well. I'm a member of the coordinating center, and that is how I'll be talking to you about my reflections on this project. So I didn't have up-to-date enrollment information. The best picture that I could give you was from a presentation that occurred in early January 2019 at the early stages of enrollment for phase 2, when we had approximately 1,200 patients who had been enrolled. But again, here and relative to that initial pie chart of Eurocentric bias in the GWAS studies, CSER in its second phase has been moderately successful, particularly in bringing in individuals of Hispanic, Latinx, ethnic identity into the study. There's also a much larger proportion of African Americans involved in CSER than was involved in CSER 2 than was involved in the first round of investigation. The aim here is not to create whole genome sequences, but basically to create exomes and genome sequence information that can actually benefit patient care, and to do so in ways that are going to promote greater access to genomic medicine and to explore barriers to genomic medicine. That is a primary objective of the CSER research study. A very big component of the RFA for the second round of CSER, in addition to more diverse patient recruitment, was a call for sustained and serious stakeholder engagement, including language from the NHGRI website describing the consortia, saying that crucial and complementary to the translational genomic efforts is a continuing focus on ethical, legal, social implications, as well as a dedicated focus on engaging stakeholders, such as professional societies, payers, regulatory agents, as well as patient groups. There has been a stakeholder engagement working group, which has met throughout regularly throughout the CSER activities, involving membership from all of the clinical recruitment sites, and they share their own local stakeholder engagement activities. The stakeholder engagement group has also promoted consortium-wide engagement activities, particularly with patient participants, such as this meeting that was held in Seattle in September 2018. These activities have been important to the work of the consortium, and they have been challenging and generated some controversy as stakeholders, particularly our patient stakeholders, have expressed needs and concerns that could not always be fully addressed in the context of the original study aims and objectives. For example, I will talk about one example here that something that I am directly involved in and quite invested in. Last summer, patient stakeholders at one of the 6 sites, CSER sites, and in light of the national reckoning around race and racism in our country in the wake of the murder of George Floyd, proposed a draft diversity and inclusion statement for joint adoption by the CSER and Ignite consortia. They brought it to us via their site's representative, and it was discussed by the scientific leadership at a steering committee call last August. There were various comments, and people recognized that it was important for a consortium like CSER to be adopting such a statement of purpose and values, and to be clear what our motivations were in doing the work that we were doing. And yet people did not like some of the language that had been proposed by the stakeholder representatives. So in September 2020, the steering committee agreed to convene an ad hoc group to discuss and revise the draft statement. Volunteer investigators were solicited. That group, which I helped lead, has met approximately monthly in the period between October and March to discuss and revise the statement, which went through a lot of discussion and numerous iterations as we tried to come to consensus on what we understood ourselves to be doing, why, and what we were willing to commit to in the face of our scientific collaborators as well as our patient stakeholders. That a final draft that took a lot of time to develop was brought to our February virtual meeting. It then received additional comments and was revised further. And just recently, indeed last week, a draft consensus statement was shared out in a stakeholder engagement discussion meeting where the national patient representatives said it was good, but it was too wordy and too intellectual, and we needed to shorten and simplify the statement. And so we are now in this important new period where we basically have come full circle back to stakeholders who basically have approved of the work that we have done to articulate our values, but want us to be clearer in our intent and to state more plainly what we are doing in CSER, why we are doing it, and how we are committing to doing this work to benefit all kinds of patient communities and to ensure that our genetic results are not misused, particularly for racist purposes. So the main take home, well, it's been a very, very interesting time, particularly in the context of the creation and editing of that diversity and inclusion statement, and has really shown me that it's hard. It is hard to meaningfully and respectfully engage diverse stakeholders. And sometimes we don't think about the research community as a stakeholder, but what we have been grappling with in CSER is trying to kind of merge and meld the perspectives and viewpoints of researchers and research participants in our shared common vision of taking an anti-racist stance in precision medicine research. And that has been challenging. It's been challenging to kind of get that right, to get the language right, and to come to a form of words and a set of commitments that everyone can sign on to. Finally, the third challenge that I would like to address is a challenge of intervening on diverse risk factors in the context of these clinical translational research studies. And for that discussion, I would like to illustrate my experience as a long-standing co-investigator with a research consortium called the Electronic Medical Records and Genomics, or eMERGE network. eMERGE also has its website. All of these consortia do. The link is provided there, and a marker paper describing the kind of past, present, and future of the eMERGE study, which gives a very nice, excuse me, a very nice overview of the work of eMERGE through the first 3 phases of the network. Was published in 2018. There has not been a update of that since the initiation of the latest phase of the eMERGE study. So the eMERGE program actually began earlier than the other two programs that I've been talking about. It actually began in its first phase in 2007. Its initial focus was to attempt to, as a proof of principle, attempt to show that GWAS could be conducted using phenotypic information that had been directly extracted from electronic health record data. And they were able to do so in phase 1. Subsequent phases have focused on taking information from GWAS and increasingly clinical sequencing and pharmacogenomic testing into clinical implementation. The newest phase, which just began last summer, is to include now explicitly 25,000 new research participants of diverse genetic ancestral background. So yes, we're in phase 4. To date, over 100,000 participants have participated in some portion of the eMERGE study. 25,000 new folks likely will be added in the context of the new study, which involves 10 clinical sites and 7 work groups. Also a very large initiative, and larger than CSER, of course, but much, much smaller than Topmed. The coordinating center for the full program is shared between Vanderbilt University and the Broad in Boston, and involves a lot of data, which are going to increasingly be going into the NHGRI equivalent of Biodata Catalyst, or the Anvil cloud data server. eMERGE 4, which is the latest phase, which is going to involve the more diverse cohort, has not yet begun recruitment. And I actually could not locate a comparable kind of public representation of the kind of racial and ethnic background for earlier phases of eMERGE, but I did find some numbers for a subset of patients who consented to participating in the sequencing part of the phase 3 eMERGE study. And this shows these data here, much more typical of what had been the case in precision medicine research initiatives until this latest kind of push to really diversify recruitment and be much more inclusive in our involvement of research participants, with a majority of participants being of white or European genetic ancestry. The next phase of the network is expected, of course, to bring in considerably more racial and ethnic diversity. So what is the issue that has come up in the context of this latest phase of eMERGE? Well, following the successful previous clinical implementation of pharmacogenetic and other kinds of more monogenic, kind of more traditional things like BRCA1 and BRCA2 Mendelian risk factors in earlier rounds, this phase of eMERGE aims to return to participants a comprehensive so-called genetic information risk assessment, or GIRA report, that encompasses a wide range of information, including clinical determinants, family health history, polygenic measures of polygenic as well as monogenic risk, as well as some social determinant information, at least which can be gleaned from the electronic health record. We can have a discussion about whether that's ever possible, but they're attempting to do it in this study. And the idea is to assess risk across these many dimensions for up to 15 different disease states or phenotypes, and to return that then to the 25,000 patients in the new phase of the study in a single report, flagging individuals as high risk who fall above a predefined threshold. It could be 95%, it might be 98%, but people who are on the population distribution of risk are at the highest end of that distribution. The details of how this is going to happen are still being actively worked out. And there's many, many questions. For example, we don't know if people on a combined risk assessment score like this who occur at the high end, if the ways in which we clinically should intervene on people at high risk are the same as the ways in which we would clinically intervene on people who have inherited a Mendelian risk factor. But for certain, a major driver of this GIRA is a polygenic risk score, which is going to be calculated from many hundreds or thousands of risk variants that are being generated in the context of genome-wide scans, like the GWAS studies that I began my presentation with. Unfortunately, those Eurocentric biases in GWAS studies have not gone away. They are still with us. And it has become abundantly clear that a polygenic risk model developed from European ancestry samples does not predict risk well in patients of other ancestral genetic backgrounds. And this is, as you could imagine, in a study which is aiming to recruit in a diverse patient cohort, a particularly thorny issue for eMERGE 4, as it is possible that the PRS that are going to go into the GIRA for some phenotypes will underperform in precisely the types of patients that we are actively soliciting to participate in the study. How best to address these considerations at enrollment as part of the informed consent process, and then during the process of result return, are currently being actively discussed and debated, informed by a range of studies by LC investigators at each of the 10 sites. Very, very important problem. Still an active conversation. It has not yet been resolved. The take home here for this study, it's really hard, really hard to build a plane while flying it. In other words, to responsibly implement risk estimates when the evidence base for those estimates is absent or incomplete. Some might argue that we should not be attempting to do so. On balance, the eMERGE program believes that it's okay that we know enough at this point, and if we're doing this in the context of a research study and appropriately caveated, this will allow us to build the evidence base, but it does put us in this very, very tricky position. And I think we're going to have to think very, very carefully about how we talk to potential research participants about the limitations of the information they might be receiving as a consequence of their participation in the eMERGE study. So, I know I'm getting close to the end of my time. So the good news is I'm getting close to the end of my slides. Where now? Where do we take these case examples that I have probably too quickly tried to step us through? And what do we think about some of these growing pains that I've observed in the course of participating in these various precision medicine research initiatives? Each, as I've indicated, are in progress, and it's really, frankly, too soon to tell how things are going to land and how things are going to end up. I do think that these illustrations point to some broader considerations for the pursuit of research with more diverse cohorts and in more diverse settings moving forward. In particular, I think it is worth returning to this depiction of thinking critically and from an ethical vantage point about the clinical translational pipeline, or as we conceived it in the context of the work of an Ethical Legal Social Implications Center of Excellence, led by my former colleague Wiley Burke at the University of Washington, a translational cycle. We need to actually be kind of thinking deliberately and reflecting specifically about the ways in which some of these early choice points in research and then in clinical implementation are influencing outcomes, and then bringing that information back to the design and subsequent the ways in which we are promoting particular categories of research. So that the lessons learned from the Topmed investigation suggests that we should be thinking very, very carefully and from the outset about how to thoughtfully manage data coming in from diverse populations, and which might be subject to different kinds of social understandings and different collections of ancillary data. We need to be thinking, as the lessons from CSER have shown us, about respectful engagement. It's been very helpful to bring stakeholders into the clinical translational research process, and it has been an interesting lesson in timing and how to think better about doing that more prospectively. And we need to be thinking very, very carefully about how we bring early fruits of clinical genomic translation into the clinical context to intervene on patients in a research setting, and how to do that work responsibly, as is indicated by the eMERGE study. And we have to be doing all of this work in continuous kind of critical ethical reflection and transparency, I think. So that's really, I see a couple of things have come up in the, maybe something has come up in the question and answer, but that's really where I wanted to get to today. I don't have any firm answers. This was mainly me kind of giving you a kind of insight into some of the struggles that I've been dealing with in the context of my on-the-ground work in the context of these programs. And as I started, and just to recap, I hope that I have at least introduced you to the issues around diversity and inclusion in the context of precision medicine research. I have walked us through how some challenges of incorporating more inclusive samples in precision medicine research contexts are panning out, and how we might have to think carefully about how we manage, engage, and intervene on diverse data, stakeholders, and risk factors. And I have began to point to, but really not come to any firm conclusions about better approaches to pursuing such research so that we can ensure that the fruits of precision medicine are equitably distributed and do not unfairly burden some participants for the benefit of others. So just to kind of to complete my presentation here by saying thanks to many, many collaborators, and past and current, including folks I've worked with in the context of the Topmed Consortium, the CSER Research Network, Electronic Medical Genomics, Medical Records and Genomics, or the eMERGE 4 study, as well as my colleagues at Columbia and UCSF, who are leading an empirical investigation of the ethics of inclusion in the context of numerous precision medicine research initiatives, including the three I've discussed today, as well as former colleagues with the UW CEER, the Center for Genomics and Healthcare Equality. I've gone over a tiny bit, but fortunately not too much. And I am eager to engage with you in your questions and discussions. Thank you so much for your attention.
Great, thank you so much, Malia, for this wonderful presentation. And yeah, on behalf of the seminar committee, which I chair, just thank you again for this opportunity to engage with you on this really interesting topic. And I should also, as Jeff did at the beginning, thank my co-committee members and Suzanne for what's been a great year working together. I'm actually going to hand over to Deborah and let Deborah have a first question, and then maybe we can go to the Q&A, and others are welcome to write more questions in the chat or Q&A. So thanks, Deborah.
Great.
So first, thank you, Malia, so much for sharing your work and reflections with us. I, of course, have approximately 1 million questions for you, but I'll start with just 2. One, I wondered if you could tell us a little bit more about, I think it was in the CSER project with regard to public engagement, your discussions around how to protect against misuse.
Mm-hmm.
What does that look like from your perspective and from the perspective of the folks you were engaging with? Yeah,
no, thank you for that. Oh, do you want to ask both questions? And then
sure. I'll put the other one, and you can decide what to answer.
All right, great. Yeah.
Sort of a technical question, and maybe it's not figured out yet because it's early days on the next version of eMERGE with the genetic information risk assessment, the JIRA. I mean, lots of questions there, but are they planning on giving sort of attributable risk to the different components? I mean, to say you have a JIRA score of whatever and not specify whether the majority of that risk is due to genetic factors or social determinants, given that those are clumped in there. And for many things, I would suspect it's mostly due to social determinants, not to genetics. Just how are they thinking about that?
All right. Yeah, those are both great questions. I'll start first with the question about protecting against research misuses in the context of CSER. That is a really, really important issue. And in the context of the diversity and inclusion statement, I mean, we certainly are gesturing in that direction, but really more tangibly, rather than the sort of performative aspects of a statement. There's been considerable work that has been going on in several different work groups over the course of this latest phase of CSER to really actively, there was an early and very full conversation about how to collect racial and ethnic identifiers and what kinds of information would be appropriate, how to classify people as medically underserved. We are now that the data have been collected and harmonized, there have been ongoing interactive conversations between the surveys and measures group and the LC and diversity group to actually talk about the ways in which basically analyses that are stratified with respect to social identifiers like race and ethnicity, better and worse ways to do that, so that information that is relevant to healthcare barriers is highlighted, but inappropriate kind of attributions that might that might imply sort of biological differences would be avoided. And the group is actively working on that issue, and there's a lot of crosstalk. We're still in the analysis phase, and so actually how it's going to play out is an open question. But I think because there has been so much interdisciplinary conversation and such goodwill and such a strong intention to avoid those harms, I believe that they will be avoided. She says, hopefully. Now, on the genetic information risk assessment, the JIRA for eMERGE, yes, all of that is still being worked out. Sometimes they are talking about it as a single risk assessment score that would be all inclusive. But for example, it is well recognized that if somebody undergoes monogenic sequencing, and this is going to be done by Invitae, and you find a Mendelian risk variant, that basically trumps everything else, and that's going to probably be at the top of the report. But how we actually think about some of the other non-genetic risk factors and whether they're going to be separate call-outs, none of that has been worked out yet. Nor do we understand how the clinicians who are going to be helping the patient participants make sense of these reports, how they will interpret and use that information. So there's a whole bunch of questions. So yeah, and none of it has been worked out yet, but there's been a lot of active discussion. Thank you for those questions.
Yep, thank you.
Great, thanks. So I just want to take one of the chat's questions here, and then if there's time, go to Karen Rothenberg, who wanted to ask a question So the first question in Q&A asks about the basis for the eMERGE study to set up a program using existing biased polygenic risk score to diverse populations and possibly return results to those participants. How will the gaps in data sets be explained to research participants, and will new PRS scores be generated using diverse data sets?
Yeah, that's it. That is the huge issue. And right now, we not only have to figure out how we're going to explain this, which I think is the secondary problem. The first problem that we're grappling with as a consortium is, are we going to report back information that we know from the get-go is not going to be as good for certain groups? And so there's a lot of very, very important work. This is primarily being led by my colleague Amir Kenny at Mount Sinai in New York City, of trying to develop polygenic risk scores, which will be portable and transferable across multiple different groups. But in certain cases, we just don't have enough data to apply. And so if a PRS score has valid information that suggests that it will perform well in 3 of 4 different racial or ethnic groups that we believe are going to be included in the study, is it good enough? Or how do we think about that when we insert caveats, either in the context of the informed consent process or later as these JIRA reports are being returned? How do we discuss population-specific limitations, and how do we explain those limitations? None of this has been worked out yet. Please know that everybody in the consortium is actively concerned with this issue. And we are, I think the worst case scenario would be that we decide that we're only going to implement JIRA in the subset of European ancestry patients in the study for whom we understand that the risk scores will work the best. Nobody wants that. But exactly what we should be doing instead is still open for discussion and debate, and we're actively working on it. And literally, as I indicated, building this plane as we're flying it.
Great, thanks so much. Let me hand it over to Karen. Karen, if you'd like to unmute, you're welcome to ask your question.
Hello, Karen.
Hi. Oh, first of all, thank you for a fantastic talk. Just fantastic. Thank you so much. My real question was, do you sleep at night?
Increasingly difficult.
You're on so many committees, and we're so grateful for that. Okay. I'm so troubled by so much of what you said, but so happy that you're at least thinking about it. So I think as it would be very helpful, I think, in the back and forth that you had with the patient participants, the stakeholders that were involved with that process. Could you tell us just a little bit more, share with us, like, what were the concerns and how did they resolve?
Right. So thank you for that, Karen. And so there's a number of different concerns that have been raised over the course of our stakeholder engagement activities in CSER. And there was actually a set of presentations recently as part of the ELSI Hub, ELSI Conversations, led by colleagues at CSER just 2 weeks ago, I think, if I'm losing all track of time, that I would strongly recommend people go and check out. And one of the bigger things that has come up is that we've been so focused on sort of access disparities at the front end, like getting people in the door to get a genetic test, that we have missed that once we get people back a genetic test result that suggests that they, or in the case of many of the CSER studies, their children have a genetic underlying cause for their medical condition, that we believe that once people are given that information, that they will be able to then access follow-on services and get the appropriate specialist help that they need. And in fact, it has proven very, very difficult. And so this is reignited for us how important it is to think about kind of the clinical handoff post a genetic test return. And then certain sites, particularly the site at UCSF, have been finding that there's kind of a need for broader social supports. Even after one moves out of the clinical domain, the people are often using genetic test information to try to access greater educational supports, for example, and having a really difficult time. And so it's really kind of broadened our sense. There are no answers for these questions because the study was not designed or set up to address these issues, but we're beginning to understand that the genetic test result is a beginning, not the end of a kind of translational process, particularly from the point of view of benefiting patients and their families.
Well, thank you for that.
Thank you.
Yeah, I
Mhm.
think we're gonna unfortunately have to wrap up. The time has run on us, and we're now reaching the top of the hour. So just to respect everyone's time, I think what we'll do is just take note of the remaining comments that have come up here in the chat and question, and maybe share them with you
Okay.
separately. But let me just say thanks again, Malia, for joining us, and thank all our participants and attendees for sticking with us throughout this seminar series. And we look forward to joining again in the fall. But thanks again, Malia.
All right. Thank you so much.